Dzień dobry Państwu, dziękuję za dołączenie na nasze webinarium poświęcone przygotowaniu bibliotek DNA do sekwencjonowania dla próbek od płynnej biopsji do analizy metagenomu. Drugą częścią naszego spotkania dzisiejszego będzie analiza wyników w programie Varsam Clinical. Niniejsze spotkanie jako Perlan organizujemy z naszymi dostawcami produktów, czyli firmą Swift Bioscience oraz firmą Safetor, sprzedawcą firmy programu Varsam Clinical. Jako Perlan mamy w swojej ofercie kompletne rozwiązania do analizy za pomocą sekwencjonowania następnej generacji, a więc oferujemy Państwu odczynniki do przygotowania bibliotek zarówno firmy Agilent, jak i Swift Bioscience, kontrolę jakości za pomocą urządzeń typu bioanalizator, tape station czy fragment analyzer, a następnie analizę wyników w programach ALISA oraz Varsam. Każde z tych rozwiązań ma swoje zalety. Dzisiaj skupimy się na rozwiązaniach od firmy Swift oraz Varsam. W skrócie telegraficznym oferta firmy Swift sprowadza się do dostarczania Państwa amplikonowych paneli, które w ciągu 2 godzin mogą dać Państwu gotową do sekwencjonowania bibliotekę. Jakość DNA pozwala na zastosowanie tych zestawów do izolatów zarówno z trudnych próbek, jak i pełnej krwi. Może być zastosowane również te zestawy do badania metylacji, do analizy transkryptomu oraz właśnie do analiz metagenomicznych. I w tym momencie analiza wyników z paneli amplikonowych może być robiona na przykład w programie Varsam Clinical. Z kolei oferta Varsam jest to możliwość analizy już pojedynczych prób, niezależnie od tego, skąd mają Państwo wyniki sekwencjonowania. Więc można przygotować biblioteki zarówno zestawami, które kupują Państwo od nas, jako firmy Perlan, ale również mogą być to wyniki uzyskane na innych platformach odczynnikowych. Olbrzymią zaletą jest to, że Varsam jest również dostępny w wersji bezpłatnej. Podejrzewam, że większość z Państwa korzysta już z tego programu do analizy wariantów. Natomiast wersja płatna firmy Varsam Clinical umożliwia Państwu automatyczną analizę wyników NGS już od plików typu FASTQ albo VCF. A teraz już zaproszę pierwszego mówcę, czyli Gerardasa Dambrauskasa z firmy Swift Bioscience. Hello, Gerardas. I would like to pass the ball to you right now. So I will stop sharing the screen and please feel free to demonstrate the solution from Swift Bioscience.
Yes, good morning, everybody. Thank you, Wojtek, for introduction, and I will start sharing my screen in a minute. Can you see the screen, Wojtek?
Not yet.
Okay. Now probably you should be able to see it.
Just make it full screen and it's perfect.
Yeah, okay, good. Yes, good morning, everybody. Once again, I'm Gerardas Dambrauskas from Swift Biosciences. We work with Perlan, with our exclusive distributor for Polish market. And today's webinar, it's in partnership with our partners for data analysis, company Varsam. So I will start with my title, as you see here on the screen. As Wojtek already introduced some of what I was going to present. So the title is Swift Amplicon Panels and Turbo. So this is our whole genome kit, which is efficient target enrichment ranging from difficult samples such as liquid biopsy, and it all the way goes to metagenomic sequencing. So a quick snapshot of the company itself. It's an American company. We're based in Michigan, Ann Arbor, and all we do is we focus on the NGS library manufacturing kits. for the compatible platforms, mainly Illumina and Ion Torrent. We do hold the wide, broad portfolio of the intellectual property that will secure the chemistry used in our kits. As it was mentioned, we focus on the cell-free DNA, FFP, PCR-free libraries, and so on. And that allows us to do, all our customers to do whole genome sequencing or target enrichment by the hybridization, which we also do have the kits. As it was mentioned, we focus on methylation analysis or methylation libraries. Amplicon sequencing, we do have a custom amplicon sequencing, and the library normalization prior pooling. So today I'm going to focus on the whole genome sequencing and the amplicon sequencing. The company is ISO certified. So this is where our portfolio, how it fits into whole genome sequencing, whether you're doing exome sequencing or targeted sequencing. So for whole genome sequencing, we use the quick and very efficient, we call it a Swift2S Turbo Kit that I'm going to focus today. And for targeted sequencing, for target enrichment, so this is a more focused approach, we use XL amplicon panels or the custom amplicon panels as well. So I will dive straight into the whole genome sequencing, and the kit that we currently do have. It's a short library preparation workflow. We call it Turbo. It's a single tube, two-step library preparation kit, which is an enzymatic target digestion. At the same time, it's a DNA repair and A-tailing. So you can see it takes about an hour of this whole step. And then the ligation of the adapter molecules, whether it's a truncated adapter or whether it's a full-length adapter, indicated here. And that will allow you to do PCR or have a PCR-free version. We use indices, combinatorial dual, unique dual, the indices that are compatible with Illumina and to some extent Ion Torrent flow cell. This is how we differentiate from our competition in terms of timing, which is clearly shown here. So the total procedure is around less than 2-hour protocol compared to our competition. Another distinguishable feature for our 2S Turbo is that independent of the quantity or concentration of your sample input, we have a very reproducible fragmentation or library fragment size, as you can see here. In comparison, we did some in-house run on our competitor, and you can see here that varying input gives you not so much reproducible insert size. And this is very, very important for your sequencing result, because different library sizes do not cluster efficiently or do not cluster evenly. So Swift went a step ahead, and not only we can have a very reproducible insert size, we can also have a varying size upon a customer's request. So we are currently the only company that does that on the market, where we can have a higher or smaller, so longer or smaller insert size upon request. So how we do it, the customer uses deceleration reagent, we call it DE, and depending on its concentration, we can drive the library towards lower end of insert sizes or higher end of insert sizes. So this is upon a request, and it's a custom-made feature that can be an add-on to the kit, especially for the metagenomics researchers. We found that they may want to work with the longer fragments that they see. Not only we can control using the DE reagent, not the library size, we can also control we can also control using the SPRI beads or the SPRI concentration. So relaxing the SPRI concentration, you can drive the libraries to a larger insert size. So those two approaches are using the deceleration reagent or the SPRI concentration sample to SPRI ratio. We can really fine-tune the wanted library insert size. We looked at the bias, and this is quite important. So GC bias, and this is quite important for the metagenomic studies, where we can see that some of the microorganisms with the high GC content tend not to be covered very well. So what we are looking here is these Packard plots, and we are looking at the line indicated by circles. So you would like to see, and this is the coverage. So you would like to see the coverage as flat as possible, closer to 1, and on the on the x-axis you see the GC content. So increasing the GC content or decreasing the GC content gives some bias from other library construction kits. Now for turbo, we looked at this very, very carefully, so you could see of a lesser bias in terms of coverage. So there's no preferential enrichment for the GC rich or GC poor regions in the libraries made by 2S Turbo. So this is another example of how we perform in terms of the sequence capture. So varying the input, so the input is going from 100 nanograms into the library construction kit. So you use Turbo to make the library, and then you use the probes. It could be from Swift, it could be from third party to do the target enrichment. And the input varies between 100 nanograms to 1 nanogram. If we look at the industry standard of what is the percentage of bases that have a 20x coverage, we perform we perform equally well as our competition. The main difference is that when you use lower inputs, and that comes from the genomic samples, or that comes from the cell-free DNA, circulating tumor DNA, where the input is always an issue, or the concentration, having enough of the sample is always an issue. So we perform with our Turbo, and this is a benchmark in our laboratory compared to our competition. So the 20x coverage doesn't drop that much if we put less of a sample compared to our competition. Just wanted to highlight this, the percentage of the duplication. This comes from the HiSeq 4000 and their flow cell. The chemistry is set up as this, so it produces quite a lot of duplications. We haven't got anything newer machine in our in-house, and I've seen some data of NovaSeq. So this is the duplication rate is dramatically reduced, probably down to about 25%. But this is inherited. This is inherited in the HiSeq machines that they produce a lot of dupes. This is our customer testimony using Swift Turbo against the competing market product. And again, they see, and this is actually their data. This is the insert size on the left, and you can see here this is a very highly reproducible insert size compared to Nextera XT in this particular case, for example. We went and did another experiment where we used the ATCC standard. This is the biodepository sample. You can get it from the company. It's a commercial, from a commercial source, and it contains those strains indicated here, and they vary at GC content. And each of the strain is at a certain proportion, and it's already indicated. So you can see here the genome proportion on the left, and these are different strains of bacteria here. And the expected genome proportion is in red, and the different inputs from into the 2S Turbo library is indicated here. So this is shows the high reproducibility and no GC bias in terms of having the genome proportion to what is expected. And as you can see here, the bacterial strains do vary from low GC content to very high GC content indicated here. So the GC content does not influence the accuracy of 2S Turbo performance in the metagenomics samples. Now I would like to switch to the amplicon panels. This is our second product where we could enrich the target by PCR. So we used to have an older version of our amplicon panels indicated here, which we still do have it on the shelf. But what we did, we recently developed the SNAP version. What that stands for is the Swift Normalized Amplicon Panel. The Normalized doesn't tell you anything at this very moment, but I will go and show what I do mean by Normalized in the later slide. So basically, if we go through the protocol, it's a 2 PCR step. It's a multiplex PCR for target enrichment with the custom designed primers or the primers that we have on the shelf, targeting specific regions, which I'll come in a second. And then it's an indexing PCR to bring on the adapters to the library. Okay, so these are the two elegant, simple steps. And then you can go and do your normal sample pooling, or you can go and do the normalize, which is embedded in the kit. What it does, it basically normalizes different libraries, up to 384 libraries. You can normalize without doing the qPCR or serial dilutions. And you end up with the pool, with one single pool that is ready to be sequenced and ready to be demultiplexed. So just a quick snapshot of the protocol without the normalized step, how we compare to our competition. So it's a two-step, one single tube. So one library gets one tube. In the competition, the library gets split between 3 tubes and so on and more. How does the normalize? And this is a unique from Swift. So imagine these are your individual libraries here. So libraries that are produced from the amplicon protocol, with the amplicon protocol that I just showed you. And it's a normalized PCR needed. So this is the last part of this attachment and indexing PCR. So that's this actually step. It's the part of the library, which you can stop here and do your own way of normalization, or you can continue in the normalize. So it doesn't really add any more steps to the extra steps to the library preparation. Then you go into the enzymatic reaction, which we call it normalize 1. Each library has to have a certain threshold, at least 6 nanomolars of the sample. In most of the cases, 6 nanomolars is very achievable in 99% of the libraries. You will have 6 nanomolars of the starting material. So imagine these are your libraries. It could be anything above 6 nanomolars. The normalized one is an enzymatic approach. that will mark a certain library concentration or certain fraction of the library in each of your libraries. And then you pool those samples and then use normalize 2. So this step here is 15-minute incubation at 37 degrees. This step is 15-minute incubation at 37 degrees. So normalize 2 will get rid of the excess of the libraries, and there is a cleanup step here which will be removed during the cleanup or the spray selection, and you will end up with just the equimolar concentration for each of the library. And as I said, it could be 384 libraries. So imagine doing 384 libraries qPCR. There's a lot in triplicates. So there's a lot of qPCR plates that you would use. And this final pool is at 2 nanomolar. So we are studying from 6 down to 2 nanomolar. The other way, we also have the kit starting from 12 nanomolar to 4 nanomolar, the final concentration of the library pool. So this is just to illustrate that your libraries, your inputs in here on the left, on the y-axis is the input concentrations. On the x-axis is the different samples and different inputs, but each library has more than 12 or more than 6 nanomolar as a threshold, above threshold. And then doing the qPCR normalization, and then demultiplexing the samples and counting the reads. So this is how the reads distribute. And the coefficient of variation between the reads is about 23.9% in this particular case. Normally, the customers get about 15% coefficient of variation with the qPCR results when they fine-tune the reaction itself or the quantitation itself. With normalize, you don't do qPCR. As I said, you use those 2 incubation steps. And when you're demultiplexing the samples indicated here, you get a coefficient of variation of 5.6%. Now, I would like to show you the real data from the University College London here in the UK. I'm based in the UK, and it's one of our reference sites here. It's a core facility. They run 30,000 reactions a year in the core facility, and they use normalize that I just described with their NEB libraries. So the normalize not just works with Swift kits, it works with the our competitors' kits. One thing to know here that normalize also works not just for amplicon libraries, but for whole genome sequencing libraries, as indicated here, which is another version. But what I wanted to show here is the coefficient of variation between the libraries, between the demultiplexed library reads stacked. So we have 20 million of reads indicated here, and there are 12 libraries demultiplexed. And you can see actually here is a very even coverage across the samples. Now, going back to the amplicon panels that I just started to introduce. So we have off-shelf panels. These are gene-based panels indicated here, and the usual panels that you see on the left. We also have a disease-based panels, which we cover. They are here on the right. And I just wanted to bring out our few best-selling panels that you see here. This is the gene list. This is what we cover. We cover the hotspot mutations indicated here. On TP53, we cover every exon and all the variants that are known in this gene. And this is the pie chart, just to break down what the coverage looks like, the pathogenic variants, just over 1,000 variants on the panel: benign 161, pathogenic or likely pathogenic. You could actually see the pie chart here. This is based on the American College of Medical Genetics or Association of Molecular Pathology. This is how we designed the panel. Now, I also wanted to highlight that most of our panels, the limit of detection or allelic frequency that you can identify using our panels is 1 or above 1%. We do have some of the panels, especially for EGFR now, that could pick up the allelic frequency of less than 1%. But it's on the shelf. It's also one of our panels that we sell. There was an upgrade to 57G pan-cancer profiling panel, which we added a few more genes to this panel. And it's important to say that our panels and our gene lists are fully compatible with Varsam, and they are routinely now analyzed with Varsam. We have a customer base increasing with our panels. So we just did the sequencing performance, or the performance of the panel indicated here with different inputs down to cell-free DNA, FFPE DNA, and we looked at the percentage on target, which is above 95% indicated here, with 10 nanogram of the input. We looked at the low frequency variant calling. As I said, we call the variants, the allelic frequency down to 1 and above. And this analysis, as I put here, is a streamlined analysis, so the data from the sequencing could be directly imported into Varsam Clinical. Not just we cover the genes, the hotspots, we cover the deletions. As indicated here, there is one common known lesion in exon 19 on chromosome 7. It's a 14 bases deletion. It's found in non-small cell lung carcinoma, as an example. Now the design process, we have the off-shelf panels. We do have the custom panels that you can request from us. So the customer provided the gene list, we provided design, we finalized with the customer the design. Swift conducts wet lab testing. So we test on the standard DNA and then the performance of the panel before we ship to the customer. Then we provide ongoing support, and we turn over to Varsam. They will take on the analysis part and the report generation part. This is the tiled amplicon coverage. So this is the exon that, as an example, shown here. And these are the amplicons, varying amplicons. They are overlapping if it's needed, if it's requested by the customer. So this is how you get the design back from our laboratory. Wanted to show some of the recent publication. One of the publications that I found quite interesting, based on our 56G oncology panel, indicated here. The sample type was the tumor-specific DNA or cell-free DNA, and they looked at using NGS and digital PCR, and this is the allele frequency. So if we look at the allele frequency of P10, which was identified with NGS sequencing of 0.6%, and they had to validate it with digital PCR, which is more sensitive than next generation sequencing. Still, they were found at the allele frequency at 1.9. So this is a very marginal, close to 1% allelic frequency. So I believe those mutations have, it makes sense to be validated by digital PCR. but something which stands for about 23% of allelic frequency, it nicely correlates with digital PCR. So I don't think that there is a need to use the digital PCR for those high allele frequencies. And there's one for the TP53 with allelic frequency that was identified 0.8%. It was confirmed with digital PCR, that is 1.4%. So as you see here, the data coming out from SWIFT panel nicely correlates with the digital PCR validation experiments. Just in summary, before I turn over to Thomas, we just discussed the SNAP panel with the normalized option included in the panel. It's a single tube, highly multiplexed assay. We can do up to 1,500 amplicons in single tube. That can be done in 2 hours. And the input is at 10 nanograms of cell-free DNA, FFPE DNA, or blood extracted DNA. Performance, we just discussed that our panels can achieve 1% or higher detection of allelic frequency. They're compatible, as I said. This is a cost-effective version, and as I mentioned, this can be analyzed with Varsam Clinical. With this in mind, I will stop here. I will turn over to Thomas. Thomas, over to you. Thank you very much.
Thank you very much, Gerardas. Maybe in the meantime, there are some questions that you would like to ask. The audience may either right now unmute yourself and ask the question, or you can also use the chat and write the questions. If not, then we can proceed with the second presentation.
Perhaps I do have a question. It's Thomas here from Varsam for Gerardas. Gerardas, could you also comment very quickly on your panels with UMIs, with unique molecular identifiers?
Yes. So we call them the high sensitivity panel. We do mostly do custom designed panels with UMIs, except one panel that we have. It's the EGFR panel that is sitting on the shelf. It's already been validated and it's been run by our customers. This is where so UMIs will help you to call allelic frequency of less than 1% with 10 nanograms of the sample input, or 0.5% with 20 nanograms of the sample input. So those panels are in works. They can be custom ordered, and they're ready to go. We use UMIs. Yeah. We call them the MIDs, molecular identifiers, but it's basically the same thing.
Right. Thank you, Gerardas.
Thank you, Thomas.
I guess if there are no other questions, I guess I can start with my part, right, by Wojtek?
Yes, please go ahead.
Let me share. Okay. Wojtek, do you see my screen?
Yes, I can. Just make it full screen and it's perfect. Yes.
All right. Good morning, everyone. This is Thomas Kusera speaking here from Safeter. As Gerardas was saying just a second ago, we start where Swift Biosciences ends, basically. And so Safeter, our company, it's a precision medicine and bioinformatics company based in Switzerland. We are on the market since 2014, and we develop tools for processing and interpretation of NGS data, specifically for clinical purposes, although we also serve research customers. So I'd like to start my presentation with bringing your attention to the challenges we are facing here when it comes to bringing NGS technology to the clinical practice. Obviously, there are many challenges out there. However, from our perspective as a precision medicine and bioinformatics company, we are focusing on 3 challenges in a particular. Challenge number 1 is the fact that the annotation data are fragmented and spread over many data resources. So I like to say that the annotation process, it's like a puzzle game, right? To get a complete picture, you need to place all the pieces of evidence in the right position. Challenge number 1, our understanding of diseases, of mechanism of diseases, it's becoming better pretty much every day. So there is basically a constant need for reevaluation and keeping your annotation data up to date. And the third big challenge we are facing here, all of us in the clinical sphere, is the lack or little standardization and consistency when it comes to the interpretation of pathogenicity. Meaning there might be 2 laboratories, for example, 2 laboratories interpreting the same variant differently, simply as a consequence of having access to different annotation data resources. So these are the 3 challenges we are addressing. So these 3 challenges are precisely the reason why we have created VARSAM, the free and open knowledge base and community. And I believe some of you have seen it before. As I'm saying, it's free and open, so you can try it anytime you like. And so what VARSAM does is, well, it does several things, but first it aggregates and cross-references annotation data resources. So we basically pull the data from over 70 data resources and bring the data in a single place called VARSAM. So in other words, these are the 70 pieces of the puzzle game. So that we have annotation data resources such as obviously GNOME-AD for population frequencies, ClinVar, expression profiles, annotation data resources for cancer samples, ICGC, PMKV, CIVIC, COSMIC, in silico predictions, conservation scores, clinically relevant data for genes, phenotypes and diseases, clinical trials, drugs, list of publications, and so on and so on. Over 70 data resources in a single place. On top of it, we keep the data always up to date. So you can be sure, you can rest assured that on VARSAM you always find the latest annotation data from all these 70 data resources. And so in other words, VARSAM, it's very solid, it's very robust knowledge base consisting of many annotation data resources, and on top of it, we implement guidelines for standardization of the interpretation process. So for example, on VARSAM, you can find, as we speak, you can find the interpretation of pathogenicity based on ACMG guidelines. So I'll get back to it in a while, but for now, I'd like to say that VARSAM's implementation of ACMG guidelines, it's very robust. It leverages the data from 70 data resources. I need to update my presentation here, I see, but it's also transparent and it's also very flexible, as you will see in a while. And we are also implementing guidelines for somatic interpretation, such as AMP, NCCN, or ASCO. AMP, it's coming in the upcoming weeks. However, VARSAM, the free platform, it's not only about annotation data resources, it's also about you, about the community around it. So VARSAM counts currently with over 250,000 users globally. So that there is a very large global community which is pretty much alive and also contribute back to Varsam knowledge base. So on Varsam, you can find publication links from our community or classifications, comments, or Varsam users even start new collaborations. And so this basically creates additional layer of annotation data, which it's unique to Varsam, and you can also leverage it for your daily work. So this is the very quick story of the free platform and community around it. Now, on top of the free platform, we have developed Varsam Clinical, which is already a professional service or a professional platform for interpretation of NGS data starting from FastQ or VCF. So with the free platform, you can look up individual variants. However, with Varsam Clinical, you can start directly from FastQ or VCF, and at the end, you get a clinical report. So Varsam Clinical, it's certified as an IDD device. It's certified as an in vitro diagnostic medical device, which means it can be used indeed in clinical settings. Our platform is also HIPAA compliant, which is relevant for our US-based customers, and our company is obviously GDPR compliant, and we are certified with ISO 13485 for quality management for medical devices. And now we are also becoming certified with ISO 27001 for information and data security. And on top of all that, we are based in Switzerland, which is country, as we know, it's a country which is very, very sensitive towards data security and data privacy. So apart from all these certifications, we have to also comply with a number of local laws in Switzerland in terms of data privacy and security. And so that means, your data are safe with us and we don't do anything with it. With Varsam Clinical, you can process your NGS data in 4 steps. First, obviously, you are supposed to upload your sequencing data to our servers. We have number of deployments for Varsam Clinical. However, most of our European customers use our own physical server in Switzerland. And so for European customers, we don't use any cloud, any commercial cloud such as Amazon or Google. Instead, we have our own physical infrastructure in data center in Switzerland. With Varsam Clinical, you can process any kind of NGS data as long as it's a human DNA. So you can process gene panels, off-the-shelf gene panels from Swift Biosciences, for example, or your custom gene panel, or exome, or genome. We support both sequencing platforms, Illumina and MGI. When it comes, for VCF files, we support Illumina and MGI. And when it comes to the VCF files, there are more options. We can accept any VCF file as long as it conforms to the standards for VCF files. So that can be, for example, VCF file from Thermo Fisher, Ion Torrent, BuzzBio, or your own VCF file as a result of the secondary analysis. Once you upload the data, you can run the pipeline. So Varsam Clinical offers a wide range of pipelines covering pretty much all the clinical use cases. We have pipelines for somatic samples, for germline samples, for pipeline for de novo variants in trios, for carrier risk screening for couples, pipeline for copy number variation, structural variation, including a pipeline for UMIS, for unique molecular identifiers, allowing detection of low frequency variants typically found in somatic samples. Varsam Clinical comes with a website interface. Obviously, it runs on our infrastructure, and you access it over the internet as if it was a regular website, which means the platform is always updated. You have always the latest annotation data available, and you don't have to worry about security, privacy, scalability issues, and so on. We take care of it. And at the end, you may generate a clinical report over which you have a complete control. So basically, you need to set up your template specifically for your workflow, and then basically you can use it for all your analysis. Template can be also fully branded according to your institutional branding policy, for example. Now, I'd like to stress out again where the challenge is here. The challenge lies in the interpretation, and I believe you would agree with me. So to do the read alignment and variant calling, in other words, the primary part, the primary analysis and secondary analysis of your NGS data is relatively simple. The real challenge indeed lies in the interpretation part of the pipeline, and that's where you need to bring as many pieces of evidence together, right? You need to get as many pieces of the puzzle game as possible in order to get a complete picture. And that's what we are really focusing on the interpretation part. Varsam Clinical covers all the steps of the pipeline, primary, secondary, and tertiary. However, our main focus here is the interpretation. Varsam Clinical, it's not a replacement for your in-house bioinformatics expertise. On contrary, it complements your bioinformatics expertise. Varsam Clinical, it's a tool, it's a productivity tool, which allows you and your bioinformatics team to work more productively, more systematically, and focus your energy where it is really needed. And together we can increase basically significantly the diagnostic yield for your patients. Here I just wanted to share with you very quickly our list of clients. So as you can see, we have quite a few clients in Europe, pretty much in all the European countries. This map again has not been updated in a while. And we have also a number of clients on the global scale in the US, Asia, Latin America, and so on. From the pricing perspective, Varsam Clinical does not have a license. Varsam Clinical charges on a per sample basis. When it comes to the FastQ files, we charge on the number of megabases in reads. And when it comes to the VCF, the price depends on the number of variants. So feel free to reach out to Wojciech and from Perlan to get the pricing sheet, as Perlan is our distributor in Poland. There are basically two ways how you can use Varsam Clinical. You can use it as the so-called standalone solution. So standalone solution, it's open for any kind of NGS data, gene panels, exomes, genomes, commercial gene panels, custom gene panels. You can upload anything you like, and we charge you on a monthly basis or quarterly basis, depending on the usage of the platform. So this is just the simple pay-as-you-go model, no commitment, no license. The other option is the so-called bundled solution. And this is where our partners, such as Swift Biosciences, come into the picture. So the bundled solution, it's a form of prepayment. And it comes with a kit, for example, from Swift Biosciences. So we have partnered up here for the bundled solution. So the bundled solution includes everything. It includes the kit, the regencies, chemicals for the library preparation, bundled with Warsaw Clinical for data interpretation and reporting. So this is a basically form of prepayment, which allows you to analyze a certain number of gene panels, for example. And the third option, in fact, there is a third option, and that's on-prem installation. We also offer on-prem installation of Warsaw Clinical, which makes sense typically only for large diagnostic centers and large hospitals with a large volume of samples. Obviously, we work with distributors. Here in Poland, we collaborate with Perlan, who is very well familiarized with the platform, and they may be able, to answer your basic support questions or questions about the use of the platform. All right, so this is my quick introduction, and now I'd like to give you a very quick introduction to Warsaw and Warsaw Clinical User Interface. So I'm trying to figure out how can I stop the screen sharing. Here it is. Oh, excuse me. Let me share it again. So now we are running out of time, I guess, but still, I'd like to show you very quickly what Warsaw and Warsaw Clinical is. To do a full demonstration, we would need at least an hour, which we don't have, obviously, and so I'm just gonna give you a very quick tour through our tools. So here you can see the free platform available at warsaw.com. I believe some of you at least have seen it before. It's the knowledge base for annotation purposes and community around it. So here on a single page, you can see a wealth of annotation data coming from all these 70 data resources. So this
Thomas, sorry to interrupt you. You may want to share your screen.
I'm not.
We can see your presentation.
Oh.
On-prem installation slide, yeah.
Now, do you see the website?
Yes.
Okay, sorry for that. It's the first time I'm using Teams, so I apologize for these issues. So so I was saying Warsaw is the free platform, the knowledge base and community around it available at warsaw.com. And it pulls the data from over 70 data resources and brings it, to a one place, to Warsaw. And so on a single page here, you can see a wealth of annotation data. This is a very, very long page, very well structured, allowing, which you can explore perhaps a bit later. For now, I'd like to tell you a little bit more very quickly about the interpretation procedure based on ACMG guidelines here. So this is a particular variant in a BRAF gene where you can see 6 ACMG criteria fired automatically based on the aggregated evidence coming from all these 70 data resources. And here you can see the explanation for every criterion, telling you in a great detail why it fired or why it did not fire. So that allows you to review every particular situation, every particular criterion, and possibly make your own judgment call. Not all the criteria can be fully automated. Certain criteria require a patient-specific information or information, for example, about for allele segregation. So in those cases, it's you who is supposed to trigger or untrigger certain criteria in line with your best knowledge. You can also specify the transcript for ACMG-based classification, which may affect, the final verdict. Or you can also specify the strength of the evidence with in line with ACMG guidelines. According for I do know, we all know that clinical laboratories rely on ClinVar a lot, which is of course very useful resource. However, according to ACMG guidelines, ClinVar is only a supporting piece of evidence. Yeah. So this is again about the puzzle game. Yeah. We shall not rely only on 2 or 3 pieces of the puzzle game. With 3 pieces, we cannot get a complete picture. We really need here to adopt more sophisticated approaches, especially when it comes to larger data sets, such as exomes and genomes. All right, now Varsom Clinical. So Varsom Clinical shares the annotation knowledge base with the free platform, with Varsom. However, it allows you, to start directly from FastQ or VCF. So first you upload your data. Here you can upload your data directly from your computer, or there is also API, application programming interface, available for every step of the analysis. So you can basically automate the whole workflow. Once you upload your data, you can start with the pipeline. You can start, from FastQ, from VCF. There is a tumor normal pipeline. There is a pipeline for trios, for carrier risk screening. You can perform a gene list analysis. There's a pipeline for copy number variation, structural variation, pipeline for UMIS, unique molecular identifiers, or MDI. So yeah, I don't know the other abbreviation. And so assuming we want to start from FastQ, we shall specify, the details of the pipeline, whether it's a germline sample, somatic sample. We shall specify the assay used for library preparation, which can be any gene panel, custom, commercial, exome, genome. For example, Swift Biosciences is already available on the platform. All the gene panels, off-the-shelf panels from Swift Bio are already available on the platform for your convenience. You shall specify the reference genome and a couple of other things. Here you can, for example, type in the patient phenotypes and diseases, and later in the process you can use these terms for variant filtering according to these patient phenotypes and diseases, for example. And then you can already start the pipeline. The analysis of a whole exome, starting from FastQ, takes about half an hour. Gene panels, depending on the size, between 5 all the way to 15, 20, half an hour, depending on the size of your data. So here I'd like to show you very quickly the results for a whole genome analysis. Here, starting from FastQ, there are always almost 5 million variants, all sorted by pathogenicity according to the ACMG guidelines. And around the variant table, you can see a wealth of annotation data broken down in tabs. Yeah, so here you can see the ACMG interpretation at the region browser, where you can see all the variants in the gene, ClinVar, Cosmic, Caviar, population frequency, somatic data resources, clinically relevant data resources such as GDC, in silico, expression profiles, in silico predictions, conservation scores, and so on and so on. All the transcript, clinically relevant data for phenotypes, genes. Where are genes? Here. HPO, human phenotype ontology, and so on and so on. Now, Varsom Clinical also allows you to set up your custom classifications. So, for example, this is a pathogenic variant according to the ACMG guidelines. No, excuse me, the first one. And however, if you don't agree with the default verdict for whatever reason, you may reclassify the variant as a benign or uncertain significance, or you can even set up your own custom classifications, such as protective or variant which needs to be discussed later, and so on. And so that's the reason why you can see here these these marks in this column here. So these tags mark your custom classifications. And the important thing here is that these classifications, custom classifications, stay with the variant. So once you upload a new sample with the same variant, you will see the same classification mark. And we can also take your manually curated list of variants. I guess you may have it in a spreadsheet, typically, and upload it privately to your account with Varsam Clinical for the purposes of variant classifications. And another really useful feature is the sample cross-referencing. Yeah, so for example, this particular variant has been found so far in a heterozygous variant and heterozygous form in 3 samples. In this analysis, another whole genome, and in another sample called the cryptic sample. Yeah, so this is something what allows you, to go back and forth between your past cases and current cases, compare allelic balances, patient phenotypes, and together with custom classifications, the sample cross-referencing allow you to increase over time your productivity and more importantly, the diagnostic yield for your patients. So this is all what we are actually talking about here, increasing the diagnostic yield for our patients. The platform comes with a number of QC reports. You can download, the coverage report, coding coverage report, region list coverage report. You can download the BAM file, VCF file. Here you can, for example, see the coding coverage report in an Excel sheet, broken down by individual genes, exon number, and sequencing depth. And so this is the place where you can identify the regions, with a low coverage, or regions that have been skipped altogether for some reason. So this sheet allows you to troubleshoot issues with assay design or issues with sequencing. So that can be very, very useful, especially when it comes to your custom gene panels, designed, for example, by Swift Biosciences. So it allows you to optimize the whole workflow. Then filters. There are quite a few filters. So let's see what's available here. You can filter your variants based on number of criteria, ACMG criteria, frequencies, ClinVar, chromosomal position, pathogenicity, zygosity, function, whether it's a 5UTR coding, splicing variant. You can set up a gene list and use it as a filter. Again, from the pricing perspective, we charge you only once. We charge you always for the initial analysis, and then you can apply all these filters, including gene lists. So you can have multiple gene lists, for example, for your exome, which are all included in the original sample price. So let's create an example. Let's see. We want to filter out, let's say, only variants with a frequency less than 1%, and only pathogenic and likely pathogenic variants. Oh, we have to give it a name. So now we have, here is the other filter. Now we have 2 filters within the filter set. And it's being applied. As you can see, the filtering works really fast. That's really important, to note. And we have built, the whole platform also keeping the performance in mind. So when it comes to genomes, right, we can have easily 5 million variants. You really need a platform which works really fast. So now you can see we are left with only 5 variants, down from 5 million. We can deactivate certain filters or bring them up again, or we can modify the whole filter set, and we can include now additional criterion based on ACMG guidelines, for example, the PP5 criterion with a strong strength of the evidence, and we are going to apply it again to our sample, and now we are left with only one variant in the variant table. So these filters are dynamic. You can play with these filters, and the results update dynamically. However, there are even more comprehensive filters called algorithmic filters. Algorithmic filters are basically a little programs we built specifically for our customers. We like to be in a close touch with our customers in order to be able to provide as much personalized services as possible, and these algorithmic filters are part of that story. So, for example, with algorithmic filters, you can analyze de novo variants in trios, or carrier risk for couples, compound heterozygous variants, segregation kind of analysis. So basically, we can encode any kind of filtering you can possibly think of. You just need to tell us how exactly you want to filter your variants, and we will develop a specific algorithmic filter for you based on that. Again, all these algorithmic filters are free of charge. I mean, the application, the initial development might be subject to charge, but ongoing use, it's already included again in the original sample price. Now, just very quickly, the reporting. So once you narrow down the list of your variants, you need to decide whether you want to report it or not. So once you make the decision to report it, you select it for export, and then you can proceed. And you can either you can either download the list of selected variants in a spreadsheet, or you can proceed and you can generate a clinical report right on the platform. So as I was saying before, this is kind of like an interactive reporting where you have a complete control over the report. So first you need to set up the template. You can customize it according to your branding policy. You can upload your logo. You can set up, the header. You can change the colors, fonts, sizes, backgrounds. You can include pictures and tables. And it can be also translated. The common elements can be also fully translated into any language. And then on the left side here, you get the list of widgets. In other words, the list of annotation, the pieces of annotation data available for a particular variant. So for example, first you can drag in the content area, and then you can drag in variant information. Then you may drag in information for the gene. Then you may drag in the list of drugs, clinical trials when it comes to somatic cases, for example. Typically you can also drag in the clinically relevant data, and then at the end list of publications. So everything is fully editable. Here you can see the content area where you can provide the custom comment for the report, and here you can see the details for the variant, AGBS term, classification of it, the transcript, exon number, zygosity, frequency, clinically relevant data for genes, list of phenotypes, list of clinical trials, list of drugs, list of publications. Oh, where are publications? Oh, and here variant references and list of publications. Everything is fully editable. So if you don't like this publication, you can take it out, and you can add additional publication here. So it's fully editable. And at the end, you can save it as a PDF file or as a doc file. So this has been obviously just a very quick introduction to Varsam Clinical. It's a complex platform with a number of sophisticated features for custom classification, sample cross-referencing, right, and so on, allowing you to increase your productivity and diagnostic yield at the end of the day. That's what we are all together trying to do, to increase the diagnostic yield and outcomes for patients. All right, so I would just very quickly conclude. Varsam Clinical, it's a complete solution for any kind of NGS data, for gene panels, off-the-shelf gene panels from Swift Bio or any other provider, exomes, genomes. There is a wide range of pipelines. There is no license, no commitment. We charge on per sample basis. And the platform is certified as an IVD device, in vitro diagnostic medical device. And you can get access to it, for example, through our partnership with Swift Biosciences as the bundled solution, which is distributed in Poland through our joint distributor through Perlan. Thank you very much, and I would be very glad to take some questions.
Thank you, Thomas. There is a question in the chat from Paulina. Is there built a pipeline tool to compare filter calls from tumor to the whole blood sequencing results?
Yes, I think you are referring to the tumor normal pairs.
Yeah, like a per analysis.
Yeah.
Okay.
It's available. Yes.
And I think that you showed that you may choose the genome version to analyze the variants, right?
Mm-hmm. You can choose from 2 reference genomes, AG19 or 38. We also annotate, all mitochondrial variants in both reference genomes.
Okay, thank you. Are there any other questions? If not, then I would like to thank everyone for your time you spent with us. And since this meeting was recorded, then I can share with you the recorded version so you can take a look at it in your free time. Thank you very much and have a very nice day.
Thank you all for taking time. Thank you, Wojtek, for organizing this. Thank you, Gerardas, and have a good day.
Thank you, everybody. Thank you. Thanks, Perlan, and thank you, Varsam.